Hello and good afternoon, everyone, and welcome to today's Lab to Impact webinar, From Bench to Market, Navigating the Complex Landscape of Drug Commercialization. We're glad that you can join us for what promises to be an engaging and insightful conversation today. I'm Brian Kelly. I am the Director of Technology Licensing and Business Development for the Center for Technology Licensing at Weill Cornell, and I will be the host for today's program, which is part of the Center of Technology Licensing's Lab to Impact, which is a virtual engagement program designed to present researchers with the latest trends, expert knowledge, and successful case studies necessary to expand the reach and impact of your research through commercialization. The Lab to Impact is organized into 4 modules, and today's program is part of our health module. We are delighted and honored to have with us today a distinguished group of panelists, each bringing unique expertise and perspectives. Before we dive into our topic, I'd like to extend a warm welcome to our panelists and ask them to introduce themselves. So maybe in no particular order, Scott, I'll maybe ask you to go first, and then Phil, Neil, and then Alex.
Sure. Thanks, Brian. So my name is Scott Tagawa. I am a professor in the Department of Medicine and adjunct appointment in the Department of Urology as well. I am an medical oncologist by training. My focus in the clinic is seeing patients with genitourinary or GU malignancies, mostly prostate, but also urothelial, kidney, testis, and other rare cancers. I run a number of different clinical trials as well, both therapeutic as well as on the diagnostic or correlative side. And my overall kind of theme has been the development of cell surface targeting therapeutics, of which one that we're going to talk about probably the most is PSMA as a target.
I guess I'm next, Brian, right?
Yes, Phil. Yeah.
First of all, I'd like to thank you, Brian, for inviting me today. It's really a pleasure to be part of this panel. My name is Phil Kantoff, and like Scott, I'm a medical oncologist, physician scientist. I spent 28 years at the Dana-Farber Cancer Institute, where I ran the genitourinary oncology program. My clinical interest was prostate cancer, GU cancers. Ran a funded laboratory for many years there, ran clinical trials, became the head of solid tumor oncology there, the division of solid tumor oncology, and also the head of clinical research. Then about 10 years ago, I moved to New York to become the chairman of medicine at Memorial Sloan Kettering, spent 6 years there. And 4 years ago or thereabouts, I returned to Boston and started Convergent Therapeutics along with my partner, Neil Bander. Neil and Scott had generated really fantastic data, which we might talk about using his antibody coupled to actinium-225. Very exciting data, and Neil asked me to join the company and start the company, which I did. So I'm now the co-founder and CEO of Convergent Therapeutics. Thank you.
Neil?
Sure. I'm Neil Bander. I was a faculty member at Weill Cornell for 40 years until I retired almost exactly 2 years ago. I was a physician scientist, surgeon scientist. My clinical interest was urologic oncology. My research interest was tumor immunology, particularly antibody targeting. I did a postdoc and fellowship in both of those areas at MSK before I moved across the street to Cornell. When I retired, or probably coincident with my retirement from Cornell, I spun 2 companies out of work that had gone on in my lab. one of which is Convergent Therapeutics, which is what we've alluded to thus far. That is a company that is developing antibody alpha particle targeting to prostate cancer by targeting the molecule called PSMA, a prostate-specific membrane antigen. I also spun another company out. Brian knows these very well because it was all done through CTL. That company is called Xenimmune, and that is developing an immunotherapeutic approach to cancer. Both of these companies are venture funded. I guess we'll talk a bit about that. And I mean, my goal as a faculty member at Cornell, beyond direct patient care, was to be able to develop targeted therapies which would have a favorable impact on patients. Alex, your turn.
Alex.
Thank you, Brian. Thank you for the invitation by the Cornell team to be here today. My name is Alex Brown. I'm the Chief Operating Officer at Convergent Therapeutics.
And
that was enough to freeze his screen.
Right. Maybe maybe we can get to Alex a little bit later, right? And Alex can join in. Let me just sort of say where we now are going to go here. So anyway, thank you for agreeing to participate and sort of share your time and insight with us, right? Today we're really going to be exploring sort of the high-level overview of the sort of 5 stages of drug development. You know, what we're anticipating over the course of the next hour is really to talk about a process that takes 20 years, so we won't be able to touch everything, and certainly in the level of detail that we're anticipating. So in terms of the stages, we're going to look at sort of the research, sort of the discovery and development stage, right? There's the preclinical research stage. There's also a clinical research stage. Then there's FDA, and the last stage is sort of FDA post-market safety monitoring. We're certainly going to touch on what some of the challenges are with respect to getting a drug to market from the company's perspective, right? And maybe some stories and lessons learned along the way. So we're first going to hear from our panelists, and then we'll try to get the audience questions towards the end of the webinar. And so in the interim, right, if you could please put your questions into the chat, and we'll try to get to them, as many of them as we can, as we proceed. So hopefully Alex will join us, right? And he's back, right? So with all that sort of let's get started, right? And maybe I'll ask Phil and Neil, right? Perhaps you could sort of speak to from your experience, right? Given your significant sort of background in academia, what are the sort of the key challenges in drug discovery in academia from your viewpoint? Phil or Neil,
Sure.
whoever wants to take that, all right?
Yeah, so Neil and I have been on both sides of the fence here, if you will, in academia and in biotech. And what I could say on the academic side is academia is really outstanding at target discovery and target validation in doing preclinical studies. It's not traditionally been where drugs are made, although there are efforts in a number of institutions to create centers that do the chemistry, to do drug screens against targets. But academia, for the most part, has not really played a big role in drug discovery. Now, having said that, we have one of the great examples of a drug emerging from academia on the panel today, work of Neil and Scott in the development of the J591 actinium conjugate we call convo-1 alpha. But for the most part, that's an unusual scenario for a drug to come out of academia. And most of the time we rely on biotech and pharma to develop drugs. So what I would say, Brian, is academia works very hard at target identification, target validation, preclinical studies, and needs to partner early on through centers like yours, Brian, to partner with pharma or biotech to develop drugs and do the pre-IND work leading to the clinical studies that are then passed back into the academic setting for the most part. Over to you, Neil.
Mr. Brown, you asked what are the difficulties?
Yeah, some of the sort of the key challenges, given your own sort of, extensive experience in the sort of the drug development space, right? That you've
I would say the number one challenge is funding. That's always difficult, and it's not just difficult in academia; it's also difficult in industry. Another issue in the academic setting is protected time, particularly if you have a clinical role. Having protected time to do research can be difficult. A third issue is finding appropriate collaborators. If you are in the translational area, which is the area that I would say that I was in, I had the advantage of having an MD, so I could see patients and literally translate what was going on in my lab to patients in the form of clinical trials. But if you are a PhD, for example, working on a translationally related project, you really need to find a clinician who shares the same interest and field of interest, so that you can work together to do that, to go through that translational process. Those are what comes to my mind.
Right. Maybe you can tell us a little bit more about, how you, were able to engage with Scott right here, and maybe Scott too. You can say from from the clinician's point of view, right? When you're looking at working with someone like Neil, right, who is, where we're trying to sort of, be more translational, right, through collaboration, and we're trying to bridge the valley of death, right, which sort of necessitates partnership, right? How that sort of collaboration came about. Maybe Neil and Scott, you can speak to both of that, right?
Well,
speak to that.
Yeah, so I'll jump in. When I first developed the J591 antibody, which really was the first antibody to the extracellular domain of PSMA, or prostate-specific membrane antigen, my immediate goal was to be able to use that antibody in clinical trials. I was able to do that. I was actually the PI on the early clinical trials. I got the IND myself. I was the PI on the trials. As I said, I would treat the patients, although I did have collaborations from GU Medical Oncology. But as you know, Brian, at Cornell and other high-level institutions, there are significant conflict of interest issues that arise. So when I first started doing these trials, I was limited by the conflicts panel to only treat 50 patients, and then I had to transfer control of the clinical trials to somebody in a different department who was not under my supervision. And that was originally to a predecessor of Scott's. But then when Scott was recruited to Cornell, which was what, about 15 years ago, Scott?
18.
18? Yeah, seems like just yesterday. But part of the reason for recruiting Scott was to take over these trials, and Scott has run the trials basically over the last 18 years. I sort of, my involvement is arm's length involvement. Scott and I would meet frequently, at least once a week. I was Scott's mentor, or one of his mentors, and we would discuss the clinical trial design and the patients and so on and so forth. But Scott was in charge of the trials and the design and the management of the patients. I wasn't able to see the patients, again, because of the conflicts issue. But it was a process that worked pretty well. I mean, Scott did a great job. I will say personally, I would have loved to be able to have had more direct involvement because I do have a clinical interest, but I do understand and accept and respect the issues of conflict of interest, so I abided by those policies. Scott, I'll turn it over to you.
Yeah, maybe Scott, you could tell us from the clinician's viewpoint, right? You know, how early are you involved in sort of looking at the potential to pursue, particularly in this instance, right? It's sort of a physician-initiated trial, right? You know, when you are the sponsor, right, of that.
Yeah, I'll just echo. I think Phil first said, and then Neil second, and I'll get third, I guess. Relatively unique. So it's a relatively unique situation, which I didn't fully recognize when I was first being recruited. But then talking to some of the people, one of the main selling points for me to move from Mount Sinai to Weill Cornell was because I could take over the development of this antibody. And by the way, there's, just for those, this parent antibody. Actually, one of the nice at the time really innovative and still innovative things that are happening now is that Neil was able to use this antibody in multiple different ways. So one is with lutetium, a beta emitter that's licensed to Telix. It's a phase 3, which we have some leadership on. There's another version that's out there, which is separate from Neil, but then also the one that is the main driver for conversion right now. So I joined at a time where this was essentially, loosely speaking, going from phase 1 to phase 2 with the initial lutetium compound. And I got to lead those initial trials, gradually also not only taking over the development, but also taking over fundraising. So much of this was funded by Neil and collaborative grants and other things. And now I've taken over the duty of getting the grants as well as other fundraising. So that was taken over an existing IND that was transferred to me, but then new INDs, there was an imaging one, and then the therapeutic one, which is now run by Convergent with has an alpha attached to it. That was collaborative preclinical work that what I'd learned over the prior 10 years or so was enough to get that into, go through the FDA and honestly teach the FDA some things because they didn't have a whole lot of experience. So as a physician sponsor rather than a pharma sponsor, they will, number one, listen to me, I think, a little bit more, and also be a little bit more lax because I'm doing this for science and patients, not so much to ultimately have something that's approved. So that's just a point that's out there for the other people that are listening in terms of academics. And then going, from a first in human phase 1 to phase 2 studies. But going beyond that, I would say it's one thing that I've learned is it's slow in academics, limited to a handful of centers. And I agree with Neil a lot of it about how much can we afford. So we did figure out how to do multi-center, but it's a small number of centers versus, some pharmaceutical companies, if there's 1,000 patient trial, they have 1,000 centers.
And What are the key data that you're looking at, Scott, in order to, convince yourself to move forward, right? Once you've had the conversation with Neil, right? And maybe you could address that. And maybe secondly, you could also sort of inform the audience a little bit about, how did you learn how to write a sort of a physician-initiated IND? How did you understand all of what is required there?
So, going back to the beginning, and Phil started with this in terms of target identification and then validation. So PSMA was New York, really, academics, Mercer Kettering and Weill Cornell. Actually, at that time, it was just Cornell. I don't think there was a while there yet. And then, developing that and proving that's a validated target. There's some people 5+ years ago that would say PSMA is not a good target. It's been out there for 20, 25 years. It can't be a good target. There's no approved products. But anyway, that having that out there, so I walked into having that, but to further develop the target, further optimize the patient population, identify, that was something that I do think that both pharma but also academics can be helpful with. Going on to your other question, in terms of what I first learned was how to, I guess, read a protocol or trial and critique it, and then how to write a trial from scratch. That was, during my fellowship training when I was at USC and learning to do that, but not necessarily on the IND part. And the IND part is partly that. So most INDs, not all, but most INDs are submitted with some idea of what's going to be the next step, which includes a protocol. But also what I did not have any know-how, and still I rely on other people's, is the chemistry section. So I think that it's possible that some people would have that entire package from chemistry to the clinical part, but more likely it's going to be a team. So developing a team of the people that are in the lab and optimizing that is something going to need toxicology. I mean, there's a lot of steps I'm skipping, but there's something going to need toxicology. And there's a lot of steps that are there, prior to actual submission and certainly approval of the IND.
Brian, could I just jump in?
Sure.
As an academic physician scientist, one of the most important things that we do is evaluate preclinical data and make decisions about, is this target, is this drug good enough to pursue as an academic? And I think in general, if the preclinical data, it does not look good, it's not going to proceed well in humans. And unfortunately, the experience has been that the majority of things that even look good in cell lines and in animal studies don't pan out in human experiments. So it's really a tough, drug development is very difficult. But making those choices as an academic translational investigator about what things to pursue, what things not to pursue, is one of the key things in becoming a successful physician scientist in academia.
That's helpful. Thanks for that, Phil. And maybe I could ask, maybe Alex and Scott, maybe, what are the sort of strategic considerations that go into designing sort of an early successful, physician, or at least a sort of a trial, right? And is there any difference from your perspective, Alex, when you're looking at sort of an industry-sponsored trial versus a physician-sponsored trial? So, and maybe Scott, for you can start, and maybe Alex, I'll ask you to sort of add to that too.
Yeah, I mean, if I were Alex, I don't know what Alex would say, but it's very clearly for a farm, especially a public company, but a private company as well, is going to be right there, have to answer to the board, and it should be mostly towards not only right. Everyone wants to help patients, everyone wants to promote science, but it's got to be towards approval in some kind of direction. Where I would love to get every drug that I think is great, including the ones that we're talking about now, out to the masses, to the patients to help them. But I also can do a trial that is going to have a bunch of embedded correlative science to learn more and to get other people some publications. And then also kind of, even though I think that maybe the most direct path towards FDA approval is this way is really interesting, is also an unmet need. So I may veer and do an additional trial rather than focusing 99 or 100% on the path to approval. So that's one of the kind of the differences, or maybe one of the key differences, I would say, in academics versus pharma.
Yeah, maybe to build on that, probably for a lot of the folks on the webinar, it's apparent that the industry is a different world from the academic world. And if you try to sort of narrow down on what are the key drivers there, it's, in the case of an academic setting, you're thinking about advancing patient care, you're thinking about advancing the field of science as a whole, you're trying to make an impact. And it's important to understand that on the industry side of things, it really boils down to you're just trying to establish risk benefit sufficiently that the FDA allows you to market a product one day. And so, unfortunately, that latter context of establishing risk benefit in order for you to market a product implies a lot higher bar of regulatory oversight and compliance. And so, as soon as you leave the hospital with a trial and you put it in the industry hands, the requirements by FDA and other bodies are so significant that it's an incredibly arduous task to conduct and complete a clinical trial. So I think that's a very big shift. And I'll be honest, there's no way that in the academic context, you could ever replicate what you're doing in industry, nor would you want to, because it would just hamstring your ability to advance care for patients, to work on the science aspect of it. So by that nature, there are two different worlds. So when is the right time to make that shift? I think, it's really when you've convinced yourself or the investigators have convinced themselves that, the data is promising. There is a drug here. This is something that needs to become available for patients. And then you might decide to take that plunge into the overly regulated or rightfully regulated world that is the industry clinical trial.
So Scott, you're doing all the interactions with the FDA, right? In terms of, how is that, how do you manage that, right? And, is that sort of how collaborative are those discussions, right? Maybe you could give us some insight into, those conversations that you would be having, right?
I think that, the word collaborative, I think that's like my viewpoint is it's more true coming from the academic than it is coming from the pharma side. So they will, I won't necessarily say, hey, you're wrong, but I would say I think that this is either from the clinic, because there may be there's different viewpoints from different reviewers, but one reviewer it'll say, no, that's not the state, it's not the unmet need in the clinic. And then one factor that we, in cancer in general, this is not just prostate, in cancer in general, when we're talking about late stage cancer, we're generally not talking about 10-year survival. We may not even be talking about 5-year survival. So why should the FDA be blocking something that might have a big benefit over 5 years, even though there is some risk maybe over 10 or 20, at least in terms of the patient population being set. So anyway, I do think there is a collaborative point of view, and that's partly why when pharma goes to the FDA, sometimes they will bring in KOLs in addition that are outside. So anyway, there is that point of view, and then as I mentioned before a little bit, the world that we're talking about right now is our therapeutic radionuclides. They're relatively new. I mean, radioactive iodine has been along for a very long time, but the development of these is relatively new, and therefore expertise everywhere. Academics, pharma, and the FDA is relatively new in being developed. So just a regular drug reviewer there's certain aspects that we should all watch out for, adverse events, etc. But not necessarily having the radioactive particle point of view, which is different than external beam radiation. So that's been a collaborative effort with me in my interactions with the FDA, probably more so than it has been with pharma.
Yeah, if I could jump in, I agree with you, but I find interactions with the FDA on the industry side to be collaborative as well. The FDA does want to see drugs developed and given to patients. The bar is higher because of the, as Alex mentioned before, or Neil did, the risk-benefit ratio has to be there. They're concerned about patient safety. But I found that so far, my interactions on the industry side have been collaborative. They want to see drugs move forward. They want to see drugs approved. They want to be helpful. So although the bar is different for industry and academia, I think they have the same general intent, and that is to be helpful.
Yeah, I would agree with both Phil and Scott. The interactions I've had with the FDA over the years have always been cordial and collaborative. We don't always agree, but it's a friendly discussion. They are clearly, excuse me, more lenient with an academic investigator. For instance, they never required us to do monkey studies, which would have been prohibitively expensive. They never required that before we went into the clinic. They would typically ask me, do you plan on developing or marketing this drug? Because that sets a different bar for them. And even though my goal was to get a drug to market, my response to them would be, no, I don't plan on doing that, because I interpreted the question in a way that I knew I wasn't going to be marketing the drug. I was going to be, at some point, turning it over to a company that would do the further development. So they gave me the leeway to get into the clinic.
And anything to add to that, Alex?
No, I mean, I think it's a reflection of the urgency, right? I mean, for Scott and Neil and Phil, when you're in that academic clinical trial, it's immediate, it's there, right? The patients are right there. And so I think the FDA, I wouldn't use the word leniency. I think they're trying to facilitate that immediate outcome, knowing that it's going to be in a smaller number of patients, but with enormous implications for the wider population. And then, as we said, it's just when you flip that over to industry, and it's just about magnitude and claims and all the downstream, that's when, I wouldn't say the regulated, we like to think of the regulated environment that I'm describing as being a bad thing. It's not. It's painful. It's a lot, and industry likes to complain about it a great deal. But the reality is the outcome of it is to design a safe and effective drug that we can consistently manufacture, distribute, market, and so forth. So, that's the gain, gaining that from the pain. So I want to make sure that we don't think of it as a horrible next step, but an important one nonetheless.
Well, it's reassuring to hear Phil say that even in today's environment, right, the FDA is, in his view, collaborative. Maybe Alex, you can give us some insight, right, into how the sort of the cadence of your meetings that you're having with the FDA, and sort of what that looks like, right?
Yeah, and for an earlier stage program, for instance, or so thinking through, let's say you have a drug that has been studied in an academic setting, and now it's moved in industry in a startup company or through a larger pharmaceutical company. What should you expect, right? Is sort of the question. And in most instances, because you're making this shift, you're looking at filing a new IND, as we sort of described earlier, and the implication of that being there's a higher bar. What does that mean? It means that unlike an investigator-sponsored IND, there's a great deal more depth and required data and information around the manufacture of the drug, in particular, and then some of the non-clinical studies that Neil mentioned. And it's substantively a lot. You're now laying the foundation for how you plan to manufacture the drug in a fashion that's scalable and consistent. So you have the interactions there, and usually, this starts with a pre-IND meeting, similar to what can happen with an investigator IND as well. So it's not dissimilar. You're reviewing with them your proposal essentially, and you're getting feedback, and that feedback is taken on board through written and in-person interactions with the FDA, and you land at your protocol, and similar to the investigator side of the equation, and you move forward. And from there, the cadence of engagement with the FDA is very much driven by the product and the kind of trial you're running. Will you have data quickly and follow up with the FDA about the next trial design and the next step? Will you be doing a lot of work in the background to develop expand or elaborate a different manufacturing approach? There's just, while the pace to seeing a drug approved is more of a marathon, there's a lot of steps along the way that, whether it's on the clinical side, on the manufacturing side, or even on the non-clinical side, you can see multiple points of engagement with the FDA. What is important, and also, to think of this holistically, is you start to layer on the engagement with regulatory bodies outside of the US, which have their own schedule, their own cadence, their own requirements. And depending on your ambition, you're speaking to regulators in Europe, in the UK, in the US, in Japan, Canada, and so forth and so forth. So that brings the focus a great deal more effort on just the regulatory engagement, depending on the speed of progress that the company chooses to take.
Actually, we've had an interesting question from the chat, right? And it's been asked sort of, when would you disclose commercial intent to the FDA?
On the academic side, or?
I think we could go with both, right? Yes. They're likely maybe different, right? But I guess it's commercial intent, right? So I guess we could, we could speak to the company, right? Maybe.
Yeah, it's an easy answer. For the commercial side, the easy answer is the moment you submit that IND request, you submit your IND. The assumption is that you're going to pursue a commercial outcome, and so everything is through that lens, which then immediately is what pushes up the bar higher on all aspects of the drug development and the clinical investigation.
I would say when assuming that there is going to be a company that's formed at that time, especially, there may be, so what we have ongoing is some academic studies are continuing on using antibody, and those are separate from the company. But I would say once the company is formed, that's the purpose of the company.
And we have another question from the chat, right? It says, is there an advantage in asking for written feedback as well as a meeting versus only written feedback sort of during the sort of pre-IND process? I'll do anybody who wants to answer that question.
Yeah, it's a little, without getting overly complicated, the answer is it's really up to the FDA what path they choose. And I wouldn't look at written versus in person as a trade-off to be concerned about. Usually, if you receive written requests from the FDA, it is either because it's not of the highest priority to you, but more likely you're receiving it because you presented the information, asked a question, and they felt that they could easily answer the question, and they provide that to you in written feedback. The FDA, ultimately, it's a group that's always underwater. They're always trying to avoid Unnecessary meetings. In-person meetings are very onerous because you have to pull people from many parts of the FDA into one conversation. So certainly for things that are lower priority, the answer is the preference is to see a written response or provide a written response. That's sort of generically how to think of it.
Mm-hmm.
I don't know, Scott, if you feel that's same representation.
Yeah, agree. I think that I'm just trying to think broadly. If there's some totally novel mechanism that hasn't been in humans before, I think having that type of a discussion, I can't think of a good example, but having that type of a discussion might be helpful. And maybe that's saying the same thing that you said, Alex, is that they can't respond in writing because they don't know what to. Usually, my experience has been the initial, I mean, sometimes zero feedback, just fine, go. Other times, it's been initial written back and forth at least once, and then as day 30 is approaching, then having a meeting. So I'd say, I don't know, less than half the time, I guess, in terms of the IND submissions, we've had a phone, a verbal conversation because it wasn't needed, but helpful the times that they happened.
Mm-hmm.
So something that might be helpful insight for the folks on the webinar who are new to this, to the regulatory component, is regardless of industry or academia, when you communicate with the FDA, you invariably, in almost every case, provide them with a summary of all the information, all the data, everything that you'd wish to discuss. The FDA does not step out of that summary to do their own research. It is just what you provide them. And so in our minds, when you think about that engagement, it's I write a dossier, I write a briefing for the FDA, they read that briefing, and it's only, they bring their personal experience, their professional experience to bear, but it's really only the substance of that briefing document that drives the conversation and the interaction. And so if you're getting a written request back, it's because you wrote a really great briefing document. If you really did not do a good job with that, or if there are questions or unknowns, there's a novel mechanism, yeah, you're going to see probably more higher likelihood of wanting an in-person meeting to discuss it further.
Mm-hmm. Another question that we have, right? Are there sort of unique challenges when you're looking at sort of either a first-in-class opportunity or a drug that has a novel mechanism of action, right? Or if you're trying to incorporate biomarkers and or companion diagnostic tests, how does that complicate the approval process?
Yes.
Yes, I guess, yeah.
The short answer is yes, all of the above. I mean, I don't think this is necessarily an industry-only concept, right? If it's new
Right.
and no one has seen it before, everyone's going to have questions, and you're going to spend a lot of time teaching. And I think that either Scott or Neil mentioned that earlier, that early on in the story of J591 and just radio therapeutics, they were teaching. And the FDA is very open to that and actually seeks that out because there's, again, no way they can know everything at all times. So I think that's the number one consideration is you're going to have to explain and you're going to have to bring people along. What is this novel construct that you're bringing to the table? Biomarker and diagnostics, or diagnostics as a whole, is the helpful way of maybe just answering that question is, in the US at least, it is a separate product. So think of it entirely as a separate path to approval from the drug. And so it just 2X's, if you will, all the considerations.
Yeah. So as we're thinking about sort of, getting a drug, as you say, to approval, right? There are things in the academic world that we don't think about, right? And that you guys have in the sort of the commercial world have unique knowledge about, right? And specific expertise, right? We don't think about things like, the sort of the challenges of regulatory approval, of manufacturing and supply issues, right? Pricing and reimbursement. Maybe you can sort of Phil and Alex, you can sort of speak to some of those challenges, right, which we may not necessarily be thinking about, right?
Right. I guess the big surprise for me moving from the academic setting to the industry side was the whole area of manufacturing. And the case of dealing with what we're trying to develop, it has unique aspects to it, because it is a just-in-time type of therapy. It's pretty complicated to put together, pretty complicated to get to patients. And these are things that I did not appreciate as much when you're in academia, because in academia, generally the drug comes from industry, and industry is taking care of the manufacturing piece of it. Another aspect that, which was alluded to before, is the whole area of funding. Funding and time are much more important in industry than they are in academia. I'm not saying funding is not important in academia, but the whole idea of milestones, getting things done in a time frame, spending the right amount of money, raising the right amount of money in order to get things done in the time frame that you want to get it done. These are things that I have learned to appreciate since going from academia to industry. Alex, you want to add on to that?
Yeah, I just taking on the other two pieces of that question. So I think in the context of a setting where you've generated clinical data in the academic setting, chances are you can move that forward in a commercial context. You know, if you think about the kind of products that are out there today, you know, we send tissue across the country for burn patients. You take cells and expand them for certain cancers like CAR Ts and put them back, modify them, put them back in a patient. So the industry will find a way if it benefits patients. I know that's maybe gross oversimplification, but it generally has been true, with a few exceptions. That is not the same. So that's from a sort of logistical, just simply can it be done for more than a handful of patients. The commercial piece is a different question. It is a real important one in some ways, because, well, what if a drug, you know, are all drugs then automatically commercially viable? And the answer is no. And at least in the US, usually the answer is, well, you have something that's interesting, but it's only slightly better than what's already out there. And so that's no different than any other commercial consideration out there for any product, regardless of medicines. If something's only slightly better, will the customer buy it? Will people want to use that in patients? And you're always up against familiarity with what people are using already. Trying something new it's, you know, for some of the academics, some of the physicians on this call, they're risk takers on behalf of their patients, but a lot of physicians are not, and they will stick with what they know. So if it's only slightly better, that's a problem. I think then there are also products that they look great, but they're just so unbelievably expensive to scale or to manufacture that they've become prohibitive as well. There's not a lot of examples of that. I think it's more on the commercial watch out is if it truly is differentiated. Is there something about it that sets it apart from what's already out there? Or, you know, is it something brand new that, you know, it doesn't have any crazy considerations around, you know, the patient has to travel to the moon and back to be administered the drug? You should be fine, at least in the US market.
Mm-hmm. Yeah, it seems to be sort of, maybe you can speak a little bit further, right, to sort of, you know, how do you really sort of face those challenges, right, of sort of, balancing innovation versus, you know, your patient benefit, right, with the increasing cost and duration of drug development, right, and how do you as a company do that, right? You know?
Yeah. Maybe in the world we're in today, even though you still see a lot of people make investments into the same type of product, you know, there might be, some of you might remember through COVID, you know, mRNA was really exciting, mRNA vaccines very exciting. And suddenly there's 100 companies developing mRNA vaccines or mRNA technologies. that doesn't seem to make a lot of sense if you think about it. But that's kind of a little bit the way that our world works. So that model has definitely been challenged recently, more so than not, because realizing, that something that's only slightly better is maybe not getting as much traction as it used to, or if it's entirely duplicative. So I think, look, when we're looking at technology now, and I think for conversion, J591 was a perfect example of this. It is very differentiated product. It has all sorts of certainly clinical and clinical aspects that we'd love to see. It's effective in patients, it's safe, but it has other incredible benefits as well, in that you only dose patients once or twice in the data that Scott and Neil generated. I mean, that's fantastic for a patient. So that's about, I get my doses and I can go home. I don't have to be treated for 2 years straight. So I think we look for those kind of tangible points of differentiation. Is this really set itself apart clinically, practically, competitively, and just from a logistical perspective, maybe. So tangible differences matter probably more than before.
Do you think there are sort of, effective models for risk sharing, right, to accelerate sort of high cost, high reward sort of drug discovery programs? How can, an academic medical center best work with sort of companies, right, to sort of better serve patients? And are there sort of new models that you Phil and you Alex would be thinking about, or potentially thinking about,
What do you think, Phil?
Go ahead, Alex, it's yours.
It's a really great question, Brian, because whenever there's a downturn, there's always opportunity, right? And
right.
We're all aware how much pressure our academic institutions and colleagues are under at the moment, for various political and economic reasons, and it's incredibly painful. But the work has to continue, right? And the science isn't going to stop, and the patients aren't going to stop knocking on the door. So I think, actually, to answer your question, is I don't think there's been a lot of great models, and very consistent models. Maybe there are some, academic institutions that have partnered with pharma and sort of, first dibs, if you will, at early research, or other types of collaborations. But I think what you're looking at now is an opportunity to do something very different. I think pharma, there was even a publication by Eli Lilly the other day, is really reconciling that they should be getting out of the research game. Research should be in an academic context. Pharma should be doing late stage clinical development and commercialization. And so maybe this is the right time to start having conversations where let's support academic research to move new drugs candidates further down the pipe. And I think that's what we saw with J591. I've never seen such a rich data set before in 30 years of my career in biotech. It's really impressive. And it helps us accelerate to move very quickly into a late stage clinical program. So maybe that is an answer where industry should invest in academia to really supplant what's been lost from government funding. And I think it could benefit patient and science tremendously.
That's
What's the model? I think we have to work on that.
all right. Okay. Well, maybe we can, right? I think as we're sort of heading towards the end of the webinar, let me just ask you back to think about sort of, looking back on your sort of your collective experience and wisdom, right? And sort of translating early drug candidates. What were some of the, maybe Phil, Neil, some of the unexpected challenges or lessons, right? That, in sort of translating and transitioning an early stage technology from the bench. I mean, any sort of some thoughts about that?
So maybe I'll start off by saying, I had a lot of praise for the FDA earlier, but regulatory aspects of drug development are pretty onerous. I'm just going to give an example of what I'm talking about. I think the FDA has patients in mind, safety in mind. There's an example, not the only example. There was a drug developed by the name of cabazitaxel in the context of prostate cancer, and it went from phase 1 to phase 3, and the dose that was picked was, I'll just throw it out there, 25 milligrams per meter squared every 3 weeks. Randomized study was done, and it showed benefit over another agent, and it was approved. But the FDA said, well, you never tested lower doses of cabazitaxel. Maybe 20 milligrams per meter squared is as good as 20, as good as 25, and safer. And they mandated that the company do 2 large randomized studies comparing 20 versus 25 after the drug had already been approved. And it did turn out that 20 milligrams per meter squared was as good as 25 and safer. So the FDA thought about that and said, well, we need to do better with regard to dose selection, schedule selection. So they came up with the idea of creating Project Optimus. Project Optimus was well-intentioned by the FDA and try to get companies in particular to arrive at the right dose. However, there was no real guidance. And many companies have been challenged by this headwind. And in our case, I think it delayed our development, trying to figure out exactly what we had to do by somewhere between 6 to 12 months. So while the FDA is patient-centric, very concerned about risk-benefit, sometimes there are some regulatory pieces that are put in front of us that are somewhat obstacles. That's my view.
Mm-hmm. I think I noticed that we are sort of, heading towards, 1 o'clock. I think we probably need to wrap this up. And I was going to leave you with one sort of final thought. I'm not sure we're going to get to this in terms of, what would be the one thing you would change that in sort of the drug discovery pipeline. Maybe we'll leave that for another day and another discussion. So as we wrap up today's sort of panel discussion, I want to take a moment to thank our incredible panelists. Thank you, Neil, Scott, Phil, and Alex. Thank you for your insight. I think you've truly helped us with our understanding and some of the challenges in drug development. Thank you for sharing your time and your perspectives with us. And to our audience, thank you for being here and for your thoughtful questions. We hope to get to more of them next time, and for engaging in this sort of important conversation. And we hope to see you all again at future CTL events. So goodbye now to everybody, and have a good rest of the day. And again, final word to the panelists. Thank you again so much for participating in this. Thank you all. Goodbye.
Brian, the answer to your last question would take an entire webinar in itself.
Right. Okay. Well, you just volunteered for it, Neil.